Hello, in this lesson we look at proteins. Proteins consist of 4 elements, namely carbon, hydrogen, oxygen, and nitrogen. And sometimes other elements such as sulfur and phosphorus may be incorporated in the protein structure, but that is not always the case. Thus, all proteins consist of the 4 elements: carbon, hydrogen, oxygen, and nitrogen. Proteins are complex molecules that are made up of long chains of smaller building blocks or molecules known as amino acids. Now, amino acids have a common structure. That is, an amino acid has a carbon atom at the center, joined to hydrogen to one end, a side group known as the R group at the other end, this is the R group, and then a carboxyl group and an amino group. Now, there are about 20 different types of amino acids that occur in nature. All amino acids are the same in the sense that they have hydrogen attached at one end, a carboxyl group, and an amino group. The variation in amino acids is due to the differences in this R group. And this is also what determines the specific properties of particular amino acid. So the R group may be as simple as a hydrogen atom, or it could be slightly more complex methyl group, or even a very large and complicated molecule that contains other elements such as sulfur and phosphorus. So it is the nature of the R group that varies from one amino acid to the next. Otherwise, they all have the amino group, the carboxyl group, and the hydrogen. Now, the carboxyl group is the one that is responsible for acidic properties of the amino acid, while the amino group is responsible for the basic properties of an amino acid. So an amino acid having both basic and acidic property is said to be amphoteric. A compound that has both acidic and basic properties is said to be amphoteric. Now, it is these amino acids that combine through condensation to form long chains known as polypeptide, and it is these polypeptides that go into the formation of protein. For instance, when 2 amino acids, so here we have amino acid 1, combines with amino acid 2, they form a molecule that has 2 amino acids known as a dipeptide. So a dipeptide consists of 2 amino acids, and the bond, the chemical bond that joins the 2 amino acids, known as the peptide bond. Now, just like in any other condensation process, water is removed in the process. So to create this peptide bond, water molecule is lost in the process. Now, when more than 2 amino acids combine, like here we have these figures here represent different amino acids, the shape and the color. So we have 1, 2, 3, 4, 5 amino acids. They combine through condensation. They form a polypeptide chain. Now, At each bond, a water molecule will be lost. So, in addition, 4 water molecules are lost in the condensation of the 5 amino acids to form the 1 polypeptide chain. Now, it is these polypeptide chains that are arranged and are combined to form the protein structure. So, a protein may consist of just 1 polypeptide chain, or may consist of several polypeptide chains joined in different patterns to form the protein. So a protein consists of 1 or more polypeptide chains. A polypeptide chain, on the other hand, consists of several amino acids. So amino acids are the building block of polypeptides. Polypeptides then constitute the proteins. A protein may be as simple as just consisting of 1 polypeptide, or it may be very complex and consisting of several polypeptide chains. Now, types of proteins. There are many different types of proteins. In fact, proteins are the most diverse, complex molecules in nature. There are so many different types. Now, the difference in the proteins stems from the following features. The difference in the large variety of proteins that occur in nature comes from the following. 1, it depends on the types of the amino acids that are present in the polypeptide. The different types of amino acids, remember there are about 20 in nature, so different types of amino acids will make up different polypeptides. The second feature is the sequence of these amino acids in the chain. Now you may have same type of amino acids in 2 different chains. Now, for example, if we use this figure, let's say in type 1, you have these sequence of amino acid, and then in another chain, we have a totally different sequence but of the same types of amino acid. So even though they have the same number and type of amino acids, the polypeptide chain 1 is very different from the polypeptide chain 2 on account of the sequence. So the difference in the sequence will provide specific properties and uniqueness to that particular polypeptide, and hence will also affect the properties of the protein. So sequence of the amino acids in the chain also matters. The third feature that determines the type of proteins is the arrangement of polypeptides in the protein structure. Now, the polypeptides may be arranged in a manner such that they form coiled and roughly spherical structures like that. So you have another chain coiling tightly, and still another one like that, so that what is formed is roughly spherical in shape. This type of proteins are known as globular proteins. The globular proteins are roughly spherical in shape, and in their structure, the polypeptides are coiled to form roughly spherical structures. Alternatively, the polypeptides can be arranged such that they run parallel to one another, like this. Now, these types of proteins in which the polypeptides run parallel along their length are said to be fibrous proteins. So as opposed to the globular proteins. In the globular proteins, the chains coil to form roughly spherical structure. Fibrous proteins, the chains run parallel to one another. Now, examples of these globular proteins, you have enzymes and some hormones. Even plasma proteins such as fibrinogen are also globular in nature. Fibrous proteins include keratin and the collagen fibers that make up the connective tissues and the bones. Keratin is found in structures such as feathers, hairs, and the nails, while collagen make up the connective tissues and parts of the bone. So the arrangement of the polypeptides in the protein structure will determine whether the protein is going to be globular in nature or fibrous in nature. Now then, the fourth feature that determines the type of proteins are the elements that are used to link the polypeptides. As mentioned earlier, these elements can have sulfur linking or phosphorus, both in the fibrous and the globular, because these chains must be held together by chemical bonds. So the formation of these chemical bonds involves the presence of additional elements which are usually sulfur or phosphorus. So some proteins may have sulfur in them, others may have phosphorus in them, as forming the link between the polypeptide chains to maintain either the globular or the fibrous structure. Now, properties of proteins. Proteins are generally insoluble in water. Proteins would not dissolve in water like the way, say, carbohydrates such as monosaccharides and disaccharides would dissolve in water. In fact, most proteins don't form true solutions in water. Instead, they form what is known as colloidal suspension. Colloidal suspension. Now, in a colloidal suspension, the particles remain suspended in the solvent, but they do not dissolve because of their large molecular size. The protein molecules are too large to dissolve, so they remain suspended. But in that state, they collectively provide a large surface area for attachment of other molecules, and it is this property that enables certain proteins to perform certain functions. The formation of the colloidal suspension, as you're going to mention when it comes to the function of the protein. So in a colloidal suspension, the protein particles do not dissolve because they are too large. They also remain suspended. They cannot sediment because they are too small. Their weight is not large enough to cause sedimentation, so they remain suspended in that state. So proteins generally do not dissolve in water to form true solution. What they form is colloidal suspension, and in particular, it is the globular proteins that form colloidal suspension. Fibrous proteins are insoluble in water. The second property is their amphoteric nature. Proteins are amphoteric, that is, they have both acidic and basic properties. Now, since each amino acid has the carboxyl group and the basic amino acid group, the proteins are acquire that amphoteric property. So this amphoteric nature enables the proteins to behave or react either as an acid or a base. If it, in the presence of an acidic substance, it behaves like a base. In the presence of a basic substance, it will behave like an acid. So for this reason, proteins play a very important role as pH buffers. Proteins help to regulate the pH of the body fluids. If there's too much acidity, their basic nature will buffer or neutralize that. If the acidity is too low, then the acidic nature will try and raise the acidic conditions. So in that way, they act as the pH buffers. Now, this property also of them being amphoteric also enables the proteins to combine with non-protein compounds. And in so doing, by combining with non-protein compounds, they form conjugated proteins. Conjugated proteins include compounds such as hemoglobin. Hemoglobin is found in red blood cells and it is used in the transportation of oxygen. Mucus, that is found in many surfaces of living organisms, is also a conjugated protein consisting of a carbohydrate and a protein. Mucus has several functions in the body, including lubrication, including absorption of unwanted products. For example, in the air that is breathed in, the mucus helps to trap the unwanted particles in the air. So this mucus is an example of conjugated protein. A third property of proteins is their sensitivity to high temperatures and extreme pH. When exposed to excess heat, usually above 40 degrees C, proteins become denatured. In the same manner, when exposed to extreme pH, that is very high acidity or very high basicity, proteins get denatured. Denaturation alters the structure of the protein molecules. Once the protein structure is altered, their chemical and physical properties change. So the proteins can no longer function normally. And that is how the denaturation comes about. Denaturing is irreversible. Hence, it permanently damages the protein in the living organisms. Functions of proteins. Proteins act as structural components. For example, proteins make up part of cells. Remember, the cell membrane, the organelle membrane, consists of proteins. Proteins, and in particular, the fibrous type. Fibrous protein makes up connective tissues, bones, tendons, muscle fibers, nails, hairs in living organisms. These parts of living organisms consists of the protein type known as the collagen and also keratin type of protein. So fibrous proteins make up structural components of body parts. The second function is as metabolic regulators. Within the cells and the body, there are certain chemical substances that control biochemical reactions. These metabolic regulators include enzymes. Now, enzymes are protein in nature and they control biochemical reactions in the cells, such as respiration. They control photosynthesis. They control synthesis of other compounds in the body. So enzymes, being protein, play a very important role in regulating and controlling many biochemical reactions. Another metabolic regulator that is protein in nature are some hormones. Hormones are compounds that regulate certain processes within the body, such as growth and functions of various organs. Many hormones are protein in nature. So, through hormones, protein do regulate many metabolic reactions in the body and even affecting the growth process. Now, during certain conditions, proteins may be used as a source of energy. For example, excess proteins, when eaten, are digested into amino acids. Amino acids cannot be stored in the body. Thus, the excess amino acids are first deaminated. It undergoes some process known as deamination to remove the amino group, and then what remains in the amino acid is oxidized to provide energy. And also, during prolonged starvation, the body proteins, the ones that make up the muscle fibers, hair, connective tissues, are used as a source of energy. They are broken down to provide energy, but this only happens when all the other sources of energy, that is carbohydrates and lipids, have been depleted. So now the body turns to structural proteins to obtain energy in order to keep vital processes such as pumping of the blood, thermoregulation going on. So under extreme conditions, proteins may be used as a source of energy. Another role of proteins in the body is as storage of nutrients. In organisms such as vertebrates that produce and lay eggs, nutrients such as albumin, which are rich in protein, or albumin in fact is a type of protein, are stored in eggs. So albumin will provide the developing embryo with all the nutrients in terms of amino acids that it requires for growth. And then proteins also do act as respiratory pigments. Respiratory pigments such as hemoglobin, present in the red blood cells of vertebrates, help to transport oxygen from the lungs to the tissues. Another pigment that is present in vertebrates is myoglobin. In particular, myoglobin is present in mammals. It is a pigment that stores oxygen in the muscles and will provide the oxygen during strenuous physical activities that requires a lot of energy to be produced. So proteins do function as respiratory pigments, mainly as hemoglobin in the blood of vertebrates and myoglobin in the muscles of mammals. Then lastly, proteins do have a protective function. In vertebrates, antibodies are produced. Antibodies destroy pathogens that have found their way into the body. In the blood of vertebrates, there are also plasma proteins such as fibrinogen and thrombin that are involved in blood clotting process to prevent excessive bleeding when the blood vessels are broken and an injury. And also, not only to prevent excessive bleeding, the clot also does prevent the entry of pathogens through the open wound. So in that way, proteins, and in particular the plasma proteins fibrinogen and thrombin, do play a role in the protection of the body.